Hey everybody! Thanks for joining our virtual talk on muscleblind and RNA localization. This is going to be a joint talk between me and Gary Bassell. Hey Gary.
Hey, Eric. Happy to be here virtually with you, although I would have rather been in Vancouver. But nonetheless, let's get started and give this joint presentation. Thank you.
My lab is very interested in these two unique, highly differentiated cell types, neurons on the left and muscle fibers on the right. Neurons have a single nucleus, yet very large projections that extend sometimes almost a meter away from the cell body. Muscle fibers, on the other hand, contain hundreds to thousands of different nuclei, essentially all in the same compartment. RNA localization and local translation are well appreciated to play important roles in synaptic plasticity and overall neuronal function. For example, beta-actin mRNA is trafficked by interacting with zip code binding protein to the tips of the growth cone. Most recently, Christine Holt's lab has shown that RNA can move along with vesicles, which are typically decorated by Rab proteins. In addition, these vesicles contain ribosomal machinery, suggesting that they contain all the components necessary for local translation. Skeletal muscle, on the other hand, is a syncytium with unique functional structures, including the neuromuscular junction, as well as myotendinous junctions in which muscle fibers are able to anchor to bone. Some RNAs have been shown to be localized to these particular functional structures. For example, RNA that encodes the acetylcholine receptor, in fact, has been shown to be localized to the NMJ. In addition, some RNAs show distributions that are sarcomeric, for example, titin RNA. And more recently, there have been studies showing that ribosomal proteins also localize to the sarcomeres in a regularly repeating fashion. Therefore, RNA localization and local translation may also play important roles in skeletal muscle. How do RNAs actually move to and stay at these locations? If one considers a typical mRNA with a poly-A tail and a cap, it's typically not naked within the cell. It's actually decorated by many different RNA binding proteins. And the particular subset of RNA binding proteins that interacts with an RNA can dictate the fate of that RNA. There are many different mechanisms by which RNAs can be localized. One is just random diffusion and anchoring. Another is differential stability, in which RNAs that might be anchored to a particular location are more stable than others. Another mechanism is directed transport, in which RNAs move along cytoskeletal filaments to reach their final destinations. There are thousands of mRNAs that are thought to be localized with specific patterns in cells. There are hundreds, if not thousands, of RNA binding proteins that can recognize these mRNAs through recognition of cis elements, for example, primary sequence or secondary structure. There are also many different Rab proteins that specify the identity of vesicles. There are also at least 40 different kinesins that have different properties and recognize different subsets of cargos. All of these components can then travel along microtubules, which can themselves be modulated by tau and MAP proteins. There's a major gap in understanding how specificity of cargos and motors is achieved. It's akin to trying to identify which passengers get on which cars or buses to go about their normal business. If trafficking is important in cells, one might imagine that mutations in motors or RNA binding proteins might cause disease. This is a short list of kinesin proteins with mutations that have been implicated in disease, along with RNA binding proteins associated with disease that have been implicated in RNA localization. For the purposes of this talk, we'll focus on the muscleblind family of RNA binding proteins implicated in myotonic dystrophy. Myotonic dystrophy is a multisystemic genetic disease that has symptoms in skeletal muscle, including muscle weakness and wasting, as well as myotonia, which is the inability to relax muscles after contraction. This disease affects the heart, causing cardiac arrhythmias, as well as the central nervous system. Fatigue and hypersomnia can affect over 90% of patients. There can also be executive functioning problems and problems with learning and memory. Myotonic dystrophy is caused by an expanded microsatellite repeat. Typically, in healthy individuals, there are fewer than 30 CTG repeats in the 3' untranslated region of the DMPK gene. In myotonic dystrophy, however, this repeat is expanded. There's a whole class of diseases caused by the expansion of short tandem repeats. More and more continue to be discovered every year. In a healthy cell, the short CTG repeat is transcribed into RNA and does not impact the free pool of muscleblind protein. In a DM1 cell, however, the repeats are much longer and end up sequestering the muscleblind proteins. Muscleblind is therefore sequestered and functionally inactivated and can't tend to its normal targets. Muscleblind proteins are well established to regulate alternative splicing. However, when we performed our clip experiments, we were surprised to see that not only did muscleblind bind introns and coding exons to regulate splicing, but it also bound extensively to 3' untranslated regions. We later showed that muscleblind actually binds to these regions in order to localize mRNAs to membrane destinations in myoblasts, And Matt Taliaferro and Chris Burge's lab later showed that muscleblind can bind to alternative last exon isoforms to localize them to distal neurites. Following on this work, there were several major questions and gaps. We wondered, are there RNA localization changes that underlie CNS symptoms in DM? In addition, what are the precise mechanisms by which muscleblind can carry these RNAs to those locations? To answer these questions, we really needed more neuroscience and RNA localization expertise. I had started going to RNA localization meetings starting in 2013 to Niagara Falls as well as 2015 to Greece. And it was at those meetings that I was fortunate to interact with experts in RNA localization. One of those experts in particular was Gary Bassell. I learned that Gary was a neurobiologist with expertise in another repeat expansion disease, Fragile X. He was also an expert in RNA localization, RNA binding proteins, and kinase motors in the context of neurons. Through talking to Gary, I found it very interesting that we had some parallels in scientific mentors. I had trained as a graduate student with Chris Burge and David Hausman at MIT, and I had heard a lot about Rob Singer through David because David and Rob had spent a lot of time together as graduate students at Brandeis. I had initially sought out David because he was one of the first to map the expanded CTG repeat in DM1 to the DMPK locus. Interestingly, in the early 90s, Rob and David worked together on first discovering the presence of CUG RNA foci, as you can see here. It turned out that Gary was actually one of Rob's first graduate students, in fact, the first graduate student to work on RNA localization. Gary tells the story best.
Rob's lab, of course, had interests in the nuclear organization of RNAs, and that was one project that I could work on, but I chose to work on RNA localization in the cytoplasm. Little did I know that 20 years later, I'd be collaborating with David Hausman's graduate student.
So Gary and I had decided to begin a collaboration. At this time, I had recently moved to the University of Florida, and Gary was at Emory University in Atlanta. We visited each other to give seminar talks as well as discuss science with lab members. The work initially began with Catherine Moss in Gary's lab as well as Tanvi Saxena in my lab, but then quickly grew to a larger group of individuals whose data we'll show you later today.
We were really excited to start this collaboration with Eric. Actually, he sent us over 20 constructs and antibodies. So we were able to actually ask two specific questions. What was the distribution of muscleblind in neurons? Were these particles perhaps modal and exhibited directed motility? We have a lot of experience to look at the distribution of RNA binding proteins in cultured cortical and hippocampal neurons. These neurons elaborate morphologically and functionally distinct types of processes, axons and dendrites. And you can see here by detection of muscleblind, MBNL1, it was present in punctate pattern. And if you zoom in, you can see that it's in both the dendrites and the axon in a granular pattern. Similarly, for muscleblind 2, we also see this punctate distribution, that it's present in the axons and the dendrites in particles. So with that behind us, we put GFP fusions into these neurons and exhibited directed motility of these MBNL granules. They moved in a processive manner, and we quantitated the behavior of these MBNL particles. They moved at rates of over a micron per second. That's consistent with microtubule and kinesin motor dependent transport. And we observed both anterograde and retrograde movements of MBNL1 and MBNL2. And so this really implicated that muscleblind may perhaps interact with specific kinesins that would motor the RNA granule to the plus ends of microtubules. And we were very intrigued in the split kinesin assay that Gary Banker's laboratory developed. This is an artificial assay that involves overexpression and creation of hybrid motors. As shown here, the BICD2 dynein adapter is fused to FKBP. The kinesin tails are fused to FRB. And these can be linked or joined by adding a small molecule into the media, allowing targeting to the minus ends of the microtubules and the accumulation of potential cargoes at the centrosome. We were able to screen over a dozen kinesin tails, and many of them did not localize muscleblind to the centrosome. Here's an example of one of those. You can see here BICD2 at the centrosome in red, KIF1B beta at the centrosome in blue. But if you look at muscleblind in green, it's distributed throughout the cytoplasm and not being targeted to the centrosome by KIF1B beta. In contrast, when we look at the related motor, KIF1B alpha, we were able to see that muscleblind was selectively targeted to the centrosome. And so by transient transfection of fluorescently fused MBNL and KIF1B alpha, we were able to see these modal particles in processive anterograde and retrograde movements by live cell imaging. Then we sought to really quantify this more precisely. And a graduate student in Eric's lab, Ryan Hildebrand, played an important role in refining the split kinesin assay, but he also uncovered a novel and selective interaction with KIF1C, which is a highly related kinesin tail to KIF1B alpha. So moving forward, we wanted to identify what RNAs might be in these granules that contain muscleblind, KIF1B alpha, or KIF1C. So Eric had actually previously published a CLIP-seq data set where he identified in brain a number of MBNL target RNAs that were decorated in their 3' UTRs. We focused on SNAP25 because it was already known that SNAP25 was having a lot of MBNL tags in the 3' UTR shown here on the top. But also it was known from Ulrich Tank's work, where he discovered that the 3' UTR was necessary and sufficient for axonal RNA localization and local protein synthesis of SNAP25, which played an important functional role in snare complex assembly and presynaptic development. By triple label fluorescence imaging by Luke Knutson, a graduate student in the lab, we were able to show that KIF1B alpha and MBNL containing granules contain endogenous SNAP25 RNA. That put us in a position to then ask what happens if you remove muscleblind from that complex, using constructs from Eric's lab to overexpress 480 CTG repeats. By fluorescence in situ hybridization, you see the RNA foci, and by double label immunofluorescence, we can show that muscleblind co-localizes to these RNA foci. This was work by a graduate student in my lab, Kun Lin, and we wanted to ask whether RNA was mislocalized. And so by fluorescence in situ hybridization, we can see SNAP25 RNA in puncta going out to the distal extremities of these mature cortical neurons. But if you look on the right at cells that have RNA foci and the muscleblind sequestration, we have a significant reduction of SNAP25 RNA in distal axons. Our lab is currently trying to investigate the functional consequences of SNAP25 RNA mislocalization. Eric's lab is focusing now on what are the molecular mechanisms on how kinesin interacts with muscleblind. Then I'm going to let him take it away and share some of the exciting research from his lab.
Thanks, Gary. So we wanted to know exactly how muscleblind associates with kinesin tails. The protein domain structure of muscleblind is quite well understood. There are two pairs of zinc fingers separated by an unstructured linker, and an unstructured C-terminal tail that contains one or two nuclear localization sequences. Using the quantitative centrosome recruitment assay, Ryan saw that full muscleblind protein can be effectively recruited by KIF1C and not by KIF1B beta. The work of Andy Berglund, my colleague, has shown that specific mutations can be made to the zinc fingers to prevent their RNA binding activity, yet not disrupt their ability to fold into zinc fingers. But upon introduction of these particular RNA binding mutations, the association was preserved. We then decided to remove the unstructured linker between the zinc fingers as well as the C-terminal tail, and to our surprise, we still could not eliminate kinesin association. We then turned to a different kind of mutation that Andy Berglund found, in which the ability of the zinc fingers to chelate zinc was abolished. These chelation mutants then showed an inability of muscleblind to be recruited to the centrosome by KIF1C. These results suggest that the zinc fingers, in a non-RNA binding dependent manner, are what are required to associate with kinesin tails. We still don't know whether this is a direct or indirect interaction, and we're pursuing that question in future studies. So in summary, the muscleblind proteins regulate alternative splicing in the nucleus, and for cargoes that get exported out of the nucleus, they associate somehow with kinesin tails in a direct or an indirect manner, such that the kinesin tails can recruit them along microtubules to distal parts of the cell. These results suggest that other RNA binding proteins might also have specific preferences for certain kinesin tails. Our work then provides a general framework for how to identify these specificities and gets us closer to the goal of truly understanding how RNA cargoes are sorted in cells. I'd like to thank a number of the people involved in this work. Ryan Hildebrandt, a graduate student in the lab who performed all the centrosome recruitment assays, Lance Dennis, another student who participated in those assays and actually initially suggested that we test KIF1C, Tom V. Saxena, who initiated this work and showed that all muscleblind isoforms can regulate RNA localization, and of course members of Gary's lab. I'd like to also thank several funding sources, the Chan Zuckerberg Initiative, Ben Barr's Career Acceleration Award, as well as several grants from the NIH.
I'd like to thank Eric Wang for recruiting me and members of my lab into this project. Katie Moss, now a postdoc at Hopkins, thanks to Gary Banker for providing unpublished reagents and sharing them with us. Luke Knudsen, a graduate student in the lab, and Chao Lin for collaborating on developing the in vitro model of myotonic dystrophy that's being continued by Kuhn Lin. Special thanks to NINDS for providing 2 multi-PI NIH grants to Eric's lab and my lab to continue to work on this exciting problem together.